Bagnato?
Yeah, sorry for my ignorance, but apoptosis is a kind of cell death with a kind of prediction, right? The cell program itself to die.
Yes.
And if I understand, you said that this is due to some level of oxidation.
Yes.
Is there a threshold where after that cells decide to die and before that decides to continue life?
Yeah, thank you. So I was just concentrating in mitochondrial oxidative alterations, and of course there are different ways by which cells can trigger apoptosis, extrinsic or intrinsic. I was mentioning a way of apoptotic death that involves the opening of what's called the redox-sensitive permeability transition pore of the mitochondrial membranes that facilitates the release of mitochondrial factors towards the cytocol, including cytochrome C, which is a master regulation of the apoptosome assembly. So you really need for the intrinsic apoptotic pathway the cytochrome C to go out and generate the apoptosome with the APAF1 and procaspase 9. And for this you need the opening of a pore. And some of this opening may be redox mediated. Of course, if the levels of oxidants are huge, the cells will die by necrosis because you need ATP for apoptosis to happen. So yes, there are different ways by which oxidants can promote proliferation at low levels, then stasis, then apoptosis, and then necrosis, depending on the ranges. So your question is welcome, and I would say that many forms of apoptosis rely on some amounts of oxidants that we promote the release of the pro-apoptotic factors. I was mentioning specifically to that type of death generated by oxidants. Of course, there are other ways of death that do not require so much initial oxidants. Many times it's just calcium influx, and secondary oxidants come to the field, to the game.
Is it possible to have a trap for free radicals?
Yes, yes. So some of the interventions we have been doing imply to restore redox homeostasis to block apoptosis. And I can say that in many models, preclinical models, there is a lot of success into that. So yes, you can interfere in different steps of the alteration, the disruption of redox homeostasis pharmacologically. We are learning this, and the mitochondrial targeted molecules seem to be a big promise to be utilized not only in vitro but in vivo because of the specificity in the site where the action is happening when mitochondria is involved in the death. So this is a prerequisite.
The next question is by Salvador Moncada.
Okay, Salvador.
No, I just want to say that once the permeability transition pore opens and things start being released from the mitochondria, that's the point of no return. The cell cannot go back once and the problem there is the general problem of free radical biology, because it's supposed to be signaling mechanisms that at a certain point become pathological. And still the conditions in which that balance takes place from physiology to pathophysiology, and what might be the situation in which the cell still goes back to normality or turns into apoptotic cell or even necrosis is not very clear at this stage. So we don't know exactly where we are in terms of where the borderline is.
Carlos Bustamante. Turn the microphone.
Yes, thank you. You mentioned the effect of radical oxidation in familial forms of ALS. Is it our understanding now about this mechanism is restricted only to familial forms, or is also suspected to be present in non-familial cases?
Thank you very much. Yeah, that's a very pertinent question, because the familial forms are the ones that have this mutation, which is utilized for the most accepted preclinical model. So how much of this translates into the sporadic forms of ALS? There is no good animal model for the sporadic forms of ALS because they don't do this disease. But the biomarkers measured in humans, in terms of oxidative biomarkers measured in the the cerebrospinal fluid and also in postmortem samples coming from human brains and human spinal cords. In both diseases are recapitulated. The familial forms usually starts a bit earlier and many times are more aggressive. Conrado may help me on that. But in terms of biomarkers of oxidation, I'd say that they are similar. So in general, there is a general agreement, I believe, that the trigger will not be the mutation of the copper zinc SOD in the sporadic forms, but there is also some redox imbalance, in addition to pro-aggregation characteristics of mitochondria, and there has been a big debate among two big lines of thinking, whether it is redox or is aggregation, and possibly redox and aggregation go together. So to me, there was never a contradiction on that, because redox imbalance promotes protein aggregation anyways. So I don't know if Conrado would like to expand on that. Okay.
We have a question by Gabriel Rabinovich.
Rafael, very nice talk.
Gracias.
So during parasite infection, for example,
Yes.
T. cruzi, so there is a balance between immunosuppression and inflammation. So what is the effect of the antioxidants that are produced by the pathogen during the global course of the disease, so during the chronicity of the disease?
Yeah, thank you. On one hand, in the acute phase, they decrease the capacity, especially the macrophages. In the case of T. cruzi, the macrophages are the first line of defense. And everything is basically decided in the first 90 minutes upon phagocytosis. If the amount of oxygens released into the phagosome are enough to really compromise the pathogen, then the acute phase is almost solved. Otherwise, after 90 minutes, the oxidative burst starts declining, and then the pathogen goes into the cytosol and starts to proliferate. Of course, that during the acute phase and during the inflammatory process of the acute phase, cytokines are released and nitric oxide production it goes up again, so there is like a second chance. But if none of these occurs and the pathogen wins because of the antioxidant capacity, in the chronic phase also the pathogen peroxyredoxins have some non-canonic actions doing immunomodulation. But this is very fresh material, and very little of that has been published. So the peroxyredoxins not only eliminate the excess of oxidants, but seems to be playing a down-regulatory role in the chronic immune response. And possibly this is part of the reason for which these parasites stay with you for over 30, 40 years. But studying the chronic phase, even in animals, is not an easy task. But I would say immunomodulation by antioxidant systems is also another side of the story. Very little done yet in terms of publishing. Thank you.
Well, Salvador.
In M1 macrophages that you have,
Yes.
do M2 macrophages also show peroxynitrite or not?
No, very, very little, in fact. So the M2 macrophages are much less efficient in generating these intermediates.
Okay.
So, yeah, that's a good point. Yeah, this phenotype is not good for forming oxidants, in fact.
Okay, thank you.
Yeah, thank you.